excited to hand it over to our next speakers now, Jonathan Gutenberg and Omar Abudayyeh. They run an amazing lab. They're at the frontier of many fields, genome editing, RNA editing, AI for protein design. Here to share with us their view on genetic agency.
Yeah, perfect. Thanks so much. Yeah, so thanks for the opportunity to speak about our lab's work today, and we hope to give you two kind of stories on how we can develop new tools, both in the molecular and computational sense, to push forward genetic agency. So this is probably something familiar to many of you, but we are actually making real progress moving towards the clinic with targeting the 1/5 of pathogenic alleles that are known to be, or 1/5 of alleles that are known to be pathogenic. This is real, of course, with the approval of Casgevy. We're seeing ex vivo therapies that are actually moving, and in vivo therapies are also moving along, although despite the recent data on the Intellia safety, there are significant progresses being made towards actually getting this in vivo. So this really makes the question of what is next, and what is the next generation of genetic medicines? I believe if you were at John's session, he probably mentioned on this, but it is insertion, and this is, I think, maybe a non-obvious fact, but really, if we look at the spectrum of mutations of disease, that not all genetic diseases, or actually majority of genetic diseases, are not driven by a single cause of mutation. There's a long tail, and diseases such as cystic fibrosis or Stargardt's have many hundreds of diseases that can cause them. And so to really create these one-and-done cures, we need to actually be able to make insertion technologies. Now, this is, of course, I think, a broad vision, but one question is, how do we make these insertion technologies compatible with how we can actually deliver right now? And I think given the context of the day, AAV is certainly quite a notable delivery vector, but how can we make these insertion technologies and other editing technologies compatible with viral vectors? And the answer to that we have been exploring is protein-free RNA writing. And so can we actually make a genome or nucleic acid editing technology that is completely protein-free? And so how do we actually do that? Well, one thing about the ideal editing technology was that we could reach and edit many tissues, not just the liver, and I think being compatible with AAV allows for this. It has a single component and a non-protein component, and that we can actually use the power of episomal expression from AAVs for a long time to avoid redosing. It can do this large insertion and also avoids permanent off-targets. And so what I'm really laying out here is being able to edit at the RNA level rather than the DNA level is a huge feature. And this actually makes us have a potentially great next generation technology, but we need to be able to do it without proteins. So how do we do this? And if you saw Arvind's talk, I believe that he may have mentioned on this as well, but we can use an approach called trans-splicing. And trans-splicing is not a new technology. It's over 25 years old, but the concept is in traditional cis-splicing, or splicing as you may be familiar with, we can essentially remove an intron and have the join the two exons. In trans-splicing, we introduce an exogenous exon that steals the exons either from the 3' or the 5' end, we can do both, to actually replace sequence and replace large amounts of sequence. So in our lab, we've been thinking and working on different ways that we can optimize and improve drastically the efficiency of this process. So this is a protein-free version, but I'll tell a bit of a story of how we got there, which is actually going through proteins. And so we initially wondered, can we actually use nucleic acid cutting proteins, such as CRISPR proteins, to improve the process of trans-splicing? And so we used a protein that we actually discovered and engineered in the lab called Cas711 to target the intron, cutting them and separating them to allow for this reaction to occur. And this kind of biasing from the cis-splicing to the trans-splicing is really the initial concept that we had. Now, how well does this work? We actually found that it worked very well. So comparing a non-targeting and a targeting CRISPR RNA, this is basically the presence or absence of that cut, we saw a 40-fold increase on a reporter transcript. And this wasn't just at the RNA level. This was at both the protein and RNA levels that we could detect by either looking at the expression of that reporter, this Gaussian luciferase, or sequencing and seeing that. So this is a great evidence that this actually worked very well. And we actually wanted to see, does this work on endogenous targets and is this safe? So we took 2 endogenous targets here, and we actually looked both, once again, at the RNA and protein level, and we saw that we could get strong correction at both. And importantly, when we do a transcriptome-wide off-target analysis, we see only that our targeted allele here, this PPIB, was actually edited in a significant way. So we have a very specific approach to actually edit. Now, once again, this is involving a protein. So really the question is, how do we actually take that to a protein-free approach? And can we start doing a protein-free method? So it was quite serendipitous. We did a simple control when we were doing a protein-based method where we took this Cas711 and we swapped it to the other strand so it cut the polyadenylation tail off of one of our cargo. We thought that this would have no effect, but we actually found that it had a massive increase in efficiency. And so we used this to target Huntington's disease, which is the first exon, and we found that we could get very high efficiency editing, which then actually mapped in vivo, so we can do this in a mouse, and we can do that at 50%, and we can even get, this is in vitro and in vivo, we can get detectable editing. How do we do this at the 3' end? We actually have been doing an additional payload with an intron to do a splicing that allows us to get very high editing at almost 15% in a protein-free system. So we're very excited about the technology. We believe it's a differentiated use case for AAV, and we're very excited about doing translation both at the 5' and 3' end on those diseases in coordination with AAV. So the next question is, how do we actually use this to discover new targets? And I'll pass that on to Omar.
Great. Thank you so much. So I'm going to change tacks a little bit about another area in our lab that we're really excited about, which is virtualizing biology through things like virtual cells. And so we got sort of drawn down this road because we have been doing a lot with CRISPR-based screening and perturb-seq, generating a lot of single-cell data based on genetic perturbations. And so we've been long interested in how do we model that data to make better predictions. And I know a lot of people hear the word virtual cell thrown around, but and there's a lot you can do with virtual cells, and it depends. Are you modeling epigenetics, RNA, proteins, all of that? Are you actually simulating down to the atoms? But we take a step back and actually try to focus on an abstraction layer of that, which is, it doesn't really matter what you put in as long as you understand what your fundamental goal is. And so for us, we're really interested in cell state, transitions. Can we, for example, identify how to bring an aged cell back to a young cell? And can we identify either genetic or small molecule interventions that can do that? And so, the reason we're really interested in perturbations is because we're, molecular tool people. We've been developing CRISPR tools and other types of tools for a long time. And so trying to better predict the effects of those tools would actually make our job a lot easier. There's been a lot of work over the past few years trying to take large language models and transformer models to do modeling of RNA, epigenetics, and so on. But fundamentally, without getting too much into this, they don't really work. So these are some of the foundation models that have come out. These are just if you do a linear regression. The graphs don't look very different, right? And so it doesn't matter how fancy your model is if you're not actually building the right architecture and collecting the right data. One of the things we think about is there isn't enough data. So if you actually compare cell data to GPTs and image models, it's multiple orders of magnitude lower. So one thing we do is we published with Feng a number of years ago a transcription factor library where you can express every transcription factor and collect single cell data. We've been expanding this across 10 new cell types, different types of primary cells and stem cells. This is mesenchymal stem cell data we recently collected for these models. Another thing is the evals are really important. So most people just predict mean gene expression. That can look really good. But we actually think there's many better evals, for example, actually predicting what genes change when you perturb a cell, what pathways change, thinking through actually what biologists care about and baking that into your model so it can do those types of predictions correctly. If you're interested in some of the architecture underlying our virtual cell, we just had a paper accepted in EUREPS that will be out soon that I would recommend checking out on archive. But generally, we're using a combination of novel, autoencoders and diffusion models to sort of make virtual cell architectures better suited for RNA-seq data. And I won't have time really to dive into the results other than to say, there's different types of perturbations you can do from, predicting new cell types you haven't seen, predicting new genes and drugs you haven't seen, to totally, unseen cells and unseen drugs, out of distribution. In general, our model can actually get, 20 to 30% accuracy in terms of differential gene expression prediction, whereas other models cannot really do out of distribution prediction. This is all zeros for SCGPT and Morph, which are two state-of-the-art models. This is just showing more data where our model is shown in blue. We can actually predict, out of the top 100 genes with 20% accuracy when you put a gene perturbation on the cell. Linear models do worse, and other existing SOTA models cannot even do this task at all. So even though they can predict mean gene expression, they cannot tell you what pathways and genes are changing. And so one of the things I think, we're seeing all these virtual cell models come out. And the real question is like, so what? Like, do they actually work? Can you actually use them? We talk a lot about, is there going to be a Move 37 move of biology, right? This is when AlphaGo from DeepMind actually did some novel move. And so for us, we think a lot about what that would be. I think it might depend on what subfield of biology you're in, but we really care about aging. And so our whole, thing that we sort of have developed in the lab are assays and experiments to find new, partial reprogramming factors. Like, can we find a single factor that could do what Yamanaka factors do and actually prove it works in a cell and in a mouse? The animation for this got scrubbed. But basically the idea is, we can screen thousands of genetic factors and small molecules for aged and young cells. So under here, the old cells should, we screen for drugs that make the old cells look young again, and we can actually find stuff. One of the molecules we found was this genocide molecule, which we found a bunch of literature for. And of course, these models do not see the literature, right? So that is pretty interesting. We're also doing this for, novel genetic factors to find ways to partially reprogram. In this case, this is muscle cells. And one last thing before I close is, none of these models can tell you why they're making the prediction. And so one limitation is how do you get mechanism out of this? And so we've been doing a lot of work actually with Google DeepMind to take all of this data, single cell data, proteomics, and actually turn it into text, actual text question and answers. Like if you perturb this cell type with this drug, what are the top 100 genes that change? And so you can create really large corpus of text data that you can then ingest via reinforcement learning into things like Gemini, which we're doing with them. And we can actually see really then much better performance on this, and you can actually see the reasoning trace, where you can see if you ask it about a gene knockout, it starts to think through what the gene knockout is, the pathways, the phenotypes it might have on the cell, and it reflects on that. And so we're really excited about virtual models of cells, tissues, humans that actually are built into language models, because then you can actually see what's going on, and the FDA likes that, right? And so with that, very excited about mixing both traditional models with reasoning models and language models to really virtualize as much of biology as possible. So thanks so much.